Hello, the structure you see here represents an intercalated disc, which is the site where two cardiac muscle cells are connected to each other. In cardiac muscle, myofibrils are arranged in a highly organized manner, which gives rise to the dark and light bands seen in this region. The dark bands, known as A bands, are mainly composed of thick filaments, whereas the light bands, called I bands, are formed by thin filaments, which are primarily made of actin. These alternating dark and light bands reflect the precise parallel arrangement of thick and thin filaments within cardiac muscle cells. Cardiac muscle cells are mononucleated and function as single individual units. Therefore, in order to act in a coordinated manner, their myofilaments must be connected to one another. This connection is achieved. Here, you can observe the structure of intercalated discs which contain numerous intercellular junctions. These junctions include adherens junctions. Adherens junctions are linked via coherens. If you look at the image, you can see that in this region, the thin filaments are connected to coherens through actin-associated proteins. The coherens of one cell can bind to the coherens of an adjacent cell, thereby allowing the filaments of one cell to connect with the filaments of the neighboring cell, ensuring coordinated contraction. Besides that, here you can also observe desmosome-like structures, where coherens are connected to intermediate filaments. The intercalated disc has both horizontal and vertical components. In the horizontal regions, you can identify gap junctions. It shows the structure of gap junction, as you can see. These are formed by connections, therefore, through these junctions, one cell can become electrically coupled to an adjacent cell. This electrical coupling allows the activity of neighboring cells to be synchronized, ensuring coordinated contraction. Intercalated disc structures can also be observed in histological slides. Here, a histological section stained specifically to highlight intercalated discs is shown from the cardiac wall. This image represents the ventricular wall, which, as you can see, is externally covered by a thin membrane called the pericardium. And most of the tissue you see here is actually the myocardium. The endocardium is not included in this particular section. In this area, you can observe cross-sections of cardiomyocytes. In cross-sections, the nucleus is centrally located, and each cell typically contains a single nucleus, although occasionally two nuclei may be seen within one cell. The pale tissue surrounding the muscle cells is the endomysial connective tissue. Within this tissue, numerous capillaries are visible, which is well explained by the high metabolic activity of the heart. If you want to observe intercalated discs, you need to look at areas where the tissue is cut in a longitudinal section. In these regions, you can see longitudinally sectioned cardiac muscle fibers along with centrally located nuclei, which allows the intercalated discs to be identified. With higher magnification in these areas, you can also observe the cross striations, which consist of dark I bands and light I bands. These striations arise from the special Parallel arrangement of thick and thin filaments. Parallel to the cross striations, you can see darker regions. These darker areas correspond to the intercalated discs, which are clearly visible with this staining method. In some cases, intercalated discs have a step-like appearance, similar to the structure I demonstrated in the three-dimensional slide. Of course, to visualize finer details, such as intercellular junctions, these structures must be studied using electron microscopy. Blood vessels are also visible in this region. This area clearly demonstrates the presence of intercalated discs.